Good morning, everyone. My name is Jacopo Franzoni. I'm, as I already said, from the University of Pisa, and I'm very excited to talk to you. And today I will show you some of the results derived from my master thesis, which was focused on the study of genotype and phenotype variation of at local scale of Dianthus virgineus populations in Tuscany. So in this study, we used an integrated approach, as many of you already presented, because when analyzing affinities and diversity in vascular plants, the integration of different data sets is really crucial, especially to assess the basis of biological variation, and especially when studying hyperdiverse plant lineages that are undergoing rapid evolutionary radiations. For instance, in the Eurasian radiation of the genus Dianthus, which is a genus that comprises around 300 species and is also widely cultivated all over the world. In the Eurasian radiation of this genus, the high diversification rate combined with frequent hybridization event produced a very difficult and unresolved systematic and taxonomic frame. More specifically, in this evolutionary lineage, the complex of Dianthus virgineus is one of the most variable of their groups, containing around 30 accepted taxa in Southern Europe, and it is characterized by an extensive morphological diversity and, of course, a complex taxonomy. In Tuscany, which was my study area, out of the around 30 taxa of this group, 3 taxa are reported, mostly on geographical and elevational basis, and sometimes with a weak support of qualitative morphological features. So, as I said before, traditionally, Dianthus sylvestris, so species sylvestris, was reported for the mountain region of Tuscany, in the Apennines and in the Appon Alps. Dianthus longicaulis was reported for the more thermophilous portion of the region, and including some of the archipelago islands. And Dianthus mauritianus was only reported for Capri Island. So, given this taxonomic background, one of the two of the main questions that arose was where if the variability of this population is distributed along some kind of gradient, and if the phenotypic variability is related to genotype variation. So, in order to answer this question, we sampled 12 populations in Tuscany, spanning from the coast of the Tuscan archipelago up to the Apennine Mountains, following a geographical and elevational gradient. We collected both flowering individuals and seeds, and then we performed the genetic, karyological, morphometric, and functional analysis in order to detect some sort of pattern of differentiation among these populations. Genetic analysis involved a genome-wide SNPs genotypification using the DD-RADSEC technique. And karyological analysis focused on counting metaphasic chromosome numbers, and the morphometric analysis were based on both multivariate and univariate analysis of measurements of vegetative and reproductive organs, made both on fresh material and herbarium specimens. And then functional analysis were focused on seed germination experiments in order to evaluate the germination responses of different populations at different temperature testings. And finally, we performed some correlation tests between the genetic, morphometric, and factual distance matrices with the geographic and elevation distances to test the presence of some statistical correlation between them. So for what concerns genetics and karyology, the genotyping of more than 600 SNPs and the chromosome counts revealed a single diploid group with 30 chromosomes. And this, let's say, lumping of population in just one group is also supported by low fixation indexes. So there is a weak population structure. And this group is genetically structured according to a geographical gradient. In fact, we can see that there are the two island populations that are separated from the continental ones, while the latter follows a geographic latitudinal gradient from southern populations on the right to northern populations on the left. So even though the results of the multivariate analysis of these 25 quantitative morphometric variables shows a marked overlapping of individuals belonging to different species and populations, is evident that this group is weakly differentiated in two main phenotypes. In fact, epinine populations are marked by smaller plant size. They have thinner leaves and few flowered inflorescences and 2 up to 6 apical scales, while Mediterranean populations are characterized by a larger plant size, broader leaves. They have many flowered inflorescences and a higher number of apical scales. And for what concerns the results of seed germination experiments, we observed that seeds of all populations are not dormant. In fact, they germinate promptly as at most temperatures tested, as we can see from this bar plot, and they prefer the cooler temperatures below 20 degrees, and these reflect the classical Mediterranean seed germination syndrome. However, lower temperatures below 10 degrees negatively affect the germination of seeds from mountain populations, and this is probably to avoid false damage during their regeneration stage. So mantle tests shows that variation, morphometric variation is not statistically correlated with the genetic fixation among populations. Meanwhile, according to Spearman correlation tests, 3 variables are negatively and significantly correlated with the increase in elevation. And these variables are the width of basal and lower stem length, the first and the third graph from the top, and the calyx length. Plants have thinner leaves and shorter calyxes at increasing elevation. On the contrary, the genetic distance is not correlated with elevation, but it is significantly correlated with the geographic distance. And finally, seed germination variation among population is not correlated with genetic fixation, but rather shows an elevation pattern. And this variation similarly highlights phenotypic plasticity of this population in seed germination responses. So, in conclusion, given these results, we can conclude that the elevation is a plausible driver of phenotypic differentiation that we observed among these populations. On the other hand, isolation by distance could play a more important role in the genetic structure of the study population. And finally, the statistical independence of phenotypic and genetic distances highlights a major role played by environmental condition, more than, of course, genetic isolation, in shaping phenotypic variation at local scales in Dianthus virgineus. So this was my thesis in synthesis. For future insights, we want to implement data analysis with some modeling. And also, thanks to the Botanical Garden of Pisa, we are common gardening the plants from the sample population that we grew from seeds in order to understand the nature of the observed morphometric differences. This work was supported by this project, which is actually supporting my PhD project that I just started, and which is focused on Dianthus virgineus, but on a larger scale. In fact, I'm studying Central Mediterranean species. And I wanted to thank all the people involved in field and lab activities, both from Pisa and Zurich. And of course, I wanted to thank you all for the attention.